All right, so here we go. We have our gene regulation notes turned on as well as the textbook. Now, from where you guys are seated in the back of the room, it's probably going to be hard for you to see my textbook. So having your own split screen really keeps you focused. And you know, the more that you are involved in class, the more that you learn. If you're sitting back and daydreaming, then you could miss 5 minutes, and then you try to catch up, and you just don't know where you are. The class is going to build on itself, so then you get behind, and then you're going to have to try to do it on your own at home, and then it doesn't make sense at home, so you don't do it. So here is perfect practice. You will get a lot of benefit out of this. And I'm trying to keep it really simple. I like that introductory video that I showed you because it gives you an overview of how new this is. This type of science has been around for 40, 50 years tops, and that sounds like a long time, but usually science goes back hundreds of years. So this is kind of recent research. And let's be aware that this is part of the upcoming test as well. So in the next two classes, we're going to have a review, and we will review this again because repetition is where you really get to understand things. It's the first time you've seen it, so this is a fire hose coming at you. But we're going to keep it simple. So look on the left on my screen. I have the textbook, Chapter 11. And we are just doing a little bit of this big idea right here, the control of gene expression. So at the beginning of class, we reviewed DNA to RNA to proteins. That's called gene expression. So the DNA code is all there, but what is going to actually turn it on or turn it off? That's the focus. We're not going to do the rest of this chapter right here. And so now, if you look over at our notes, this picture that you see in your notes, this is your key to practice. And if you can talk your way through this picture by next Wednesday's quiz, Tuesday or Wednesday, depending on your class, then you're ready. So there's a lot going on in this picture, so let's just review what we can see. Everybody see the nucleus in purple? And we've learned before that that's where DNA is, and that's where messenger RNA is. So that's where transcription occurs. So that's why they have the word transcription up in the nucleus. Then we know that messenger RNA leaves the nucleus and gets translated. That's why in the cytoplasm it says the word translation. And it's translating from messenger RNA to protein. So you see this pipeline right here? There are, I think it was 9. 2, 4, 6, 8, 9. There are 9 controls that can shut off the expression of the gene from the very beginning so that transcription doesn't even happen. And then if you have messenger RNA, we can shut it down in 4 ways down here so that you never get to a protein. The building blocks of proteins, these amino acids, they're in short supply, so they might have to be recycled and also held onto until you really need the protein. So we're going to go over these 5 basic ways that the cell is going to control it. So you see the numbers, 1, 2, 3, 4, 5? And then the notes are 1, 2, 3, 4, 5. So do you see the overall scope of that? After we go through these 5 types of controlling genes, you're going to be running a simulation. So you're going to be going to a website. This is a virtual lab. These notes right here that we're going to take are part of a lab grade. This is a 35%. And this is worth 20 points. So this is worth, what, 75% of next week's test? So it has the same value on it. So take good notes here and practice these notes because this is part of your quiz. So do you remember Hank used the word methylation? So we're going to explain how the transcription can be turned off from the very start. And this is where you're inherited some of these methylations. So that video really gives you the importance of living a good, healthy life for yourself. But if you want to have healthy children too, then some of this is passed on. It's just kind of mind-blowing that we have genetics on top of our DNA. And so where can we find that in the textbook? Chapter 11, section 2. So that's what that number means. So with your textbook now, you can go to the second page. And I'm in chapter 11, section 2, and there's a picture that Hank was talking to you about. He even had a price tag, didn't he? So he had like a price tag hanging off of this like that. Another tag hanging off of that. So that's epigenetics, and that's what methylation is. So over here in your notes, you're going to write explain methylation. Methylation is a molecule that attaches onto DNA and closes it. Verbal notes. Methylation is a chemical that attaches onto DNA and keeps it closed. When DNA is closed up like this and wound around these proteins, you can't get at translating it into messenger RNA. So now over here in your notes, you would want to write something like it closes the DNA up with a chemical tag, and it's going to stop transcription because when DNA is closed up, it can't be accessed. For those of you who are interested, and you're going to be taking chemistry class, in your textbook, in section 2, it has chemical modifications, and you inherit this epigenetics. It's right here. I highlighted it. Methyl groups are CH3. So over in your notes, you could write, because I just wrote a chemical tag, and the chemical tag is CH3. This note-taking style that I've been showing you with a textbook on one side and your notes on the other is how you can study at home. Would you consider taking notes yourself by looking over 5 pages? Even though it's not graded, would you view that as extra? Or would you view it as part of your homework because you need to work a little bit more to understand it? If you don't have curiosity, end of story, right? You just don't, you won't do anything. So that's the number one thing in science and learning, that you're curious to explore things you don't know and then expand your knowledge, and it might lead to more questions and more curiosity. Don't you do that with video games? You know you want to get to level, 75? And you're at level 35 and you can't get past it, so you go do research. You go to YouTube. You used to have to buy these little quick help guides, to help you get through a game, or you call up your friend. So you're curious about getting past it. So just realize that if you want to improve on your academics, stay curious. Don't give up easy. All right. Right next to that section in the textbook, explained acetyl groups. Did anybody find it? Yeah?
Isn't it kind of the opposite of methylation? So it opens the
Great. Yep.
chromatin structure.
And it's in that same paragraph still. Section 2 in chapter 11. You should follow along. Right here it says that it's another chemical and it opens up the DNA. And it's COCH3. I'm not going to test you on the chemistry of it. You're going to get plenty of chemistry later. So if you want to keep it generic and say it's a chemical that opens up DNA, you're golden. So now back over here to the notes. Was it a CH2 or 3? 3? Yeah, that makes sense because carbon needs 4 bonds. So one of them is to oxygen and then 3 hydrogens. So if you were doing this on your own and you were just copying down the book definition over here, A on homework. Don't know what you're writing down? Hate to say it, D on test, right? So you have to practice your notes in the right way. If you think you're bad at tests and you are destined to be bad at all tests, no, it's just you're not using the tools correctly, maybe making it too complicated, maybe being distracted while you're doing notes. All right, let's pick up some speed here and realize that we're still in this area of how do we turn off transcription or how does it get turned on to make messenger RNA. So it's activation proteins, and there's a picture for that. It's in section 2. Excuse me, section 3. I'm in the textbook, section 3. And take a look at the entire picture, and it even labels it. These are proteins, and they're needed to activate the gene. So here is DNA, and it is getting ready to make RNA. Without these purple and green pieces there before the gene, you're not going to have the main builder come in. Do you remember the builder in your DNA replication? You opened up and you showed DNA polymerase coming in. Well, there is something that has to happen first. You have to have helpers. So here's the answer simplified. I probably could type this neater than writing it, so sorry about that. So RNA polymerase is going to come in here to make messenger RNA, but before that can happen, you need to have proteins that are going to do some pretty cool things. It's going to take DNA and fold it over on itself like this. It's almost, what do you call that when you go like that?
A dab.
So in order to make the DNA go like that, proteins have to grab this guy and then fold over like that. And this is the only time that messenger RNA can be made in humans or eukaryotic cells. So yeah, isn't that a good visual? So you got to go like that in order for messenger RNA to be made. And that is with the help of these activation proteins. Isn't the picture really telling the story? Did any of you draw a little picture? So you just draw a little. Here's a quick story. Over the last weekend, I was talking to my friend who travels a lot in his RV. And when you're on a long trip, sometimes you don't make it to an RV resort. So you can pull into a Walmart, you can do some shopping, you can get some food, and then you could just rest in the parking lot for a few hours and then head out. So most Walmarts will let you do that. But there wasn't a clear sign, and there was like 5 or 10 RVs there. They woke up in the morning and they had a boot on their car. You know what that is? The police lock up the wheel so it can't roll. There was even someone who was waiting for their spouse to get off of work, and he just was like before 5 o'clock in the morning, and he fell asleep. And they came and they put a boot on. They didn't say, "Hey, wake up! You're not allowed to be here." They just like silently put boots on the car, and they wouldn't take it off until they paid the fine for overnight parking in the parking lot. So I'm telling you the story for this next one right here under number 1. Explain how a repressor protein turns off genes. It's a boot on the gene. Here's the picture in red. It's going to lock on right before the gene and not allow RNA polymerase to do its job. So if you want to write in quotations, it's a boot on DNA. So RNA polymerase cannot attach. And then if RNA polymerase cannot attach, then it is going to block the gene and in effect turn it off. Understand that the normal state for genes is to be turned off. So this is a common thing where you have a lock on it. And how do you get a lock to unlock? You need a key. And the key in this picture is going to be, I lost my spot. This is a picture in section number 1. So it's like that. Here is the boot on the DNA, and another molecule is going to have to come in here like a key, and then it makes it change its shape, and the repressor falls off of the gene and now can be transcribed. So aren't you getting an appreciation for how wonderfully made we are with all this chemistry working without you even knowing about it? There are all these chemicals that are on your DNA to open it, to close it, to boot it, to put things to activate it. So this is the number 1 reason that your genes are either turned off or turned on. It's at transcription. This is the main valve that you see right up here on that picture in your notes. Let's move on. Question number 2, or reason number 2, mechanism number 2, and we are now in section 4 in your notes. Do you remember that when messenger RNA is made, up in this picture, that it is a raw messenger RNA that doesn't work? It has to be processed. Do you remember us talking about how it needs a cap and a tail? It needs to have a little aglet on your shoelace. It has to be finished on the corners. It also should be sliced and edited so that it doesn't have extra stuff. So here is the raw messenger RNA that was transcribed, but it has to have these things added to it. It's not even called messenger RNA until it's done like that. Otherwise, it's just called RNA transcript, coming from transcription. So the answer to question number 2, right in here in your notes, is it needs to be processed to become RNA. Oops, excuse me, mRNA. Without this processing, the whole process can stop and you're not going to make a protein. Yeah, it's hard for me to make text boxes here. Start typing. Good. Yeah. I'm going to give you a short answer for number 3. How is messenger RNA translation regulated? You need to have proteins present just like we did in transcription. They're also needed at the ribosome for everything to attach properly. So I've given you a quick answer right there. And in that, there always has to be additional chemistry available to say, yes, proceed or do not proceed. There are 9 places for a checkpoint to occur. And I really like this next one. Once you have messenger RNA, it could be transcribed. And you're going to see this when you run your simulation. And we're just about there. MicroRNA. RNA is usually really long. Let's take a really small piece of it and call it a micro piece of RNA. And here's a picture of it. It is in section 6. Section 6 in your textbook has a ribosome right here. It has the purple messenger RNA. And then it has a small piece of microRNA. What does this microRNA do? It complementary base pairs with it, and now it's kind of like a boot. It's blocking it, but it does more than that. It slices and dices it, and you see the graphic. Messenger RNA was nice and ready to be translated, and along comes another attachment, and it just breaks it apart. So this is a major way of destroying messenger RNA, because if you have the recipe right there, somebody comes along and they just rip it apart and shred it in a shredder, and then they go, you can't make it anymore because your instructions are gone. So it's kind of an effective way with small pieces of messenger RNA attaching to it. It's called microRNA. So we can simplify that. How good are you at making things simple from the complex? A small piece of RNA attaches to messenger RNA and destroys it. Chops it up. Number 4, we have allowed the protein to be made. Transcription and translation occurred. We have 2 more chances to ruin the protein party. I'll show you a picture of it.
Yeah, I was short.
Yes, a picture. All right, let's finish up these notes, and here we go. This purple right here is a polypeptide. That's a protein. And proteins, I don't know if you remember, there was an earlier question a month ago. It said, how many times does a protein have to fold in order for it to work? A minimum of 3, usually 4. So that's like 1, 2, 3, and then 4. So when a protein is first made, it's just a long strand of amino acids, right? So it's just raw, and it needs to be processed. The way it gets processed is by folding, and sometimes it can't fold right without being cut in certain places. So it's showing 2 cleavage sites, and now you have pieces that come together in a different formation. So, can you put that in your own words? How is a new protein, it's so raw that it isn't even folded yet and in the right shape? Proteins only work if they're in the right shape, and pH and temperature can really affect their shape. So it's best if you write something down instead of waiting for your teachers to always tell you what to write down. So if you are in the driver's seat instead of in the back seat, then you see more and you learn more. Doesn't have. You use the. Yeah, I can't remember what they do for that. Let's forget that. Just pour the extra into those other ones. I'm going to write raw proteins need editing and folding maybe 4 times in order to work. So if your protein is not going to be finished, then it's not, you may have made it, but if you don't polish it up, it's not going to work. All right, bonus points. What organelle in the cell will do this? What is the organelle that will repackage and add things to protein before it's sent out of the cell? It's not ribosomes. Ribosomes make the protein. The endoplasmic reticulum carries a protein to this organelle. What organelle am I talking about? It looks like a stack of pancakes. It has 2 words called the blank apparatus. Golgi apparatus. So there's an organelle that will do this. So if it was made and doesn't make its way over there, the cell can still control. It might, in response to you eating spicy food, it might be trying to make an enzyme to break it down. But then if it's all done and the food isn't there anymore, it would go, oh, sorry, don't need this anymore. And then it would just destroy it. So question number 5, how are proteins eventually broken down? It might get to the point where it has folded it properly, but at the last second it goes, oops, sorry, don't need it. Now we need this protein. So it goes, all right, I'm going to break this one. I'm going to grab the amino acids, have them available for transfer RNA. And so it's quite simply right here. To mark a protein for destruction, it's going to add a molecule to the protein. It's almost like a blinking beacon that's going to go, oh no, I've been tagged, destroy me, destroy me. It's like, oh, they're going to get me. And then it's going to be destroyed by enzymes. So how can we say that in your words so that you can understand it? If you just write down what's in the book, it usually doesn't serve you very well. A marker, like a beacon, will be attached to proteins, marking them for destruction. Marking them for destruction by an enzyme. Remember I said this is like a fire hose coming at you today? So there was a lot of information and you may have gotten it the first time, but you need to practice it a little bit. Otherwise, this may have not made any sense to you, so you should listen to the recording of our class. You should open up the textbook. You should come to office hour with another student. Instead of just sitting there for recess, you could be prepping for your quiz. Your quiz is next Wednesday. If you wait till Tuesday night, you're on the track for getting a D on the quiz. So your destiny is in your own hands by studying and practicing earlier than that. With 25 classes left, you want to make sure that you're hitting a good average. So if you want your grade to end where you are right now, keep doing what you're doing. If you want to have a different result, try something different. Come on into office hour. Let's work on your preparation and study skills. All right, at the very last page right here, it talks about this virtual lab you're going to do. And some of you can finish this in 5 minutes, because that's all the time we have. But others might need a little bit more time. I will extend the due date to the end of the day, so you're able to do that if you'd like, since we did go a little bit longer. You have to do this. Here is what the game looks like. You have to do 3 genes. You have to run through 3 screens. On each screen, there's 1 gene. Here's the gene. After you do this, you'll either go to the previous gene or to the next gene, and notice that it says it right down here. It'll say gene 1, gene 2, gene 3. Here's your toolbox. Remember we talked about activation proteins? They have to be in place before RNA polymerase can come in and transcribe the gene. So you're going to drag these players onto the screen in the right spot, and you can't fail. Once you put your finger on it and go over here, a spot on the screen is going to blink in the shape of the object that you need to place it, so it'll get you placing it in the right spot. But you have to do it in the right order. So if you're turning the gene on, you have to have the positive transcription factor. If you need to turn the gene off, you'll have a negative transcription factor. Do you remember that microRNA that will destroy messenger RNA? That's Pac-Man right here. If you take Pac-Man out and move it over to messenger RNA, it chomps it up and destroys it. So once you have messenger RNA and ribosomes on the scene, you know that translation can occur. Translation is going to make the protein pop right out of the ribosome, as we know it does. And now you're going to grab that protein and you are going to drag it into the scorecard right here. You start with 0 in these 3 proteins, and you have to get them to at least 5. It doesn't take long. Once you're done with this protein, these other 2 proteins are on the other screen. So you go to previous gene or next gene. And it's a really cool visual of what we just talked about with transcription factors, with destroyers. So this is how your genes are turned on and off with these 9 different mechanisms. So I'll come around and help you to get started. I recommend that you get started just so that when you pick it up again later, you can finish this in a matter of minutes.